All right, so we're officially on air now. So we're going to take some time to introduce ourselves to you guys. So my name's Catherine, and I am a second-year graduate student here at UNC.
And I'm Nicole, and I'm also a second-year graduate student in UNC.
And first we're just going to kind of explain a couple things about ourselves, starting with how we got interested in science. So for me, that actually began when I was in high school. I had an excellent chemistry teacher in my sophomore year, and even though I didn't think I was the best at chemistry, she told me that if I really loved it, that I should stick with it and try as hard as I can, and that's what I did. And so I'll let Nicole explain how she got excited about science.
So I was exactly your age when I decided that I wanted to be a scientist. I was in 8th grade, and basically I just took a basic science class, and I just was really interested in science. I wasn't necessarily extremely good at it, but it was something that I was really interested in and that I really, really liked. And so I just stuck with it. And so after 8th grade, I went to high school and took a bunch of science classes and found that in particular I liked chemistry the best. And so that's what I went forward with.
Cool. So we're both in graduate school now, and so to kind of explain to y'all what that means is we went to college, and then graduate school is something you can choose to do after college. And so that's basically a very specialized field of study, so we've chosen chemistry. And our day-to-day lives, we do research all day. Some of us teach as well, some labs. And so I personally chose graduate school because I was thinking about medical school, but I quickly realized I couldn't deal with blood. And so after some research during the summer, I decided to pursue graduate school.
And I decided to go to graduate school because, well, I went to high school, took a bunch of classes, realized I liked chemistry, then went to college and got a degree in chemistry, and then I wanted to keep going because I just felt like I love science so much. I just kept wanting to learn more, and so I went to graduate school, which is a step further. And so now me and Catherine are on the way to becoming real scientists.
And so just a little bit about the research we do now. I work in a lab that's looking at trying to make devices to generate hydrogen energy. And so it's very focused on looking for new ways to create new sources of energy. And after graduate school, I actually want to go back and teach, just because I really felt like I've been impacted by some really good teachers, and I think that I will enjoy doing that for the rest of my life. So.
Yeah. So now the research that I do in graduate school is I develop devices that allow us to study just a single cell. So monitoring how a single cell behaves and how it becomes, for example, mutated to become something like a cancer cell. So ultimately, the goal is to understand the cell cycle and how we can better treat certain diseases and things like that.
And so we want to pause right now to see if you guys have any questions about all that information we just shared. If not, we'll go on to some background.
Any questions?
Any questions? I don't see any questions.
Okay. Okay.
All right. So now we're going to screen share with you a PowerPoint presentation that's going to kind of give you guys a little bit of background on the microscope we're going to be looking at, as well as comparing it to a light microscope, which you've all probably heard about. So Nicole's actually going to give you a little bit of detail about a light microscope and explain what that is.
Right, so all of you in class should have already used a light microscope before, right? So we have this place at the top where you look with your eye through the lens, and then you look down at your sample. And your sample is on a stage, and we're using light in order to see our sample. And so the light is coming from the bottom, hitting our sample, and then allowing us to see that image with our eye, right? And then we're able to adjust the focus to make things less blurry, so we can actually see what we're looking at by adjusting these little focus knobs.
And so to talk about the microscope we're using, what we're using is called a scanning electron microscope, and that's shown over here. And what's on the left is a light microscope that Nicole just talked about. And to give you kind of an idea, we've flipped this image, so we've actually flipped this microscope, so our eye is now down here. And so in a scanning electron microscope, we, instead of using like our eye as a detector, we actually use electrons to detect our species on our stage down here. And so we can still use focusing elements, so we can focus this beam of electrons similarly to how we do with light. The only difference is that our eye, again, is not involved. We just use these beam of electrons to focus on our image. And then the computer will actually generate images, and that's what we will be showing you guys today of these species, of our specimens. And so I want to talk a little bit about length scales. So the human eye can see things pretty large, so we can see each other. We can see our own hands, we can see our fingers, and we can see human hair. However, that's kind of where our resolving power of the human eye stops. Light microscopes, we can investigate the human hair, and so your own human hair is about 100 microns thick. And then we can also look at red blood cells and also bacteria with a light microscope. So with a light microscope, we're really in the micron range. And then with an electron microscope, we can go down even farther in length scale. So we can see things like virus particles, DNA molecules, glucose molecules, and even a single atom. And so that's all the way down to the nanometer range, which is much smaller. And so throughout our session today, I'll be kind of referring to the length scales mainly in reference to your hair. So I'll talk about how small we are looking at in comparison to your hair. And so we'll also point out our scale bar as well, so you can see how small we're looking. Okay, and so are there any questions about any of that material?
I don't see any.
Okay. All right, so now we're going to show you the microscope. And so we're going to have to move around here a little bit so we can show it to you.
All right.
Okay. And so this right here is our microscope. So first thing you should notice is that it's fairly larger than your standard light microscope. Our electron beam is actually right here, and so this is kind of like the light in a light microscope. And then what you notice is we have this chamber here, and this chamber is under vacuum, so I can't open it right now to show you the inside, but basically we put our samples under vacuum so we can get really good images of them. And then I just kind of wanted to give you an idea of some sample stubs, so what we load our samples on. And so you know in a light microscope you use a stage. You put your samples on a stage. But in ESEM we use these little sample stubs. And so you can see how small they are. And at the top we have this copper tape where we'll actually stick our sample on. And then we can set this inside the ESEM and then put it under vacuum. And so this is kind of what our stage looks like here. Okay, yeah, so are there any questions about the microscope or how we load samples, how much this costs?
How much does it cost?
Yeah, how much does it cost?
Full price, probably $500,000.
So about $500,000 sitting right here. So yeah, people who use this have to be trained, and so we have experts here that will train us on how to use this instrument so that we don't break it because it is very expensive. Okay, so I think we're going to start analyzing the samples you guys sent us.
Yeah.
So we'll get situated here.
Want to replug it?
Okay. And so now, to change your screen view, if you will click on our microscope view to make that be the big image.
Got it?
Yeah. And so before we kind of zoom in on what you're seeing, we're going to actually talk through all the views you're looking at. So in the upper left quadrant is where we're actually going to be taking images. Okay. In the lower right quadrant, what you'll see is actually our sample stage. And so you can kind of see all the samples we've loaded inside. Mm-hmm. And to give you guys an idea of what samples we chose, we put in the ladybug, the bumblebee, the fly, and then the orange bug thing that I haven't been able to identify. And so those are kind of the samples that we are able to choose from. And also inside this chamber, you can see, again, the electron beam right here. And so we kind of want to let you guys choose what sample we look at first. So again, your options are the fly, bumblebee, ladybug, or the orange bug.
Who says ladybug?
Ladybug? Okay. Okay.
We'll start with ladybug.
So Nicole's going to move our stage over to the ladybug, and then you should be able to start to see some things. Okay. So what you're seeing is the most zoomed out we can go with this image. And so, again, because we're not working with a light microscope, we can't really see colors, but we can kind of see the ladybug's features still with this microscope. And so in the lower right-hand corner, again, we have our scale bar. And so that's about 2 millimeters in length that we're looking at. And so we're going to zoom in on some features on this ladybug here. And so ladybugs, actually, the hard casing around the ladybug, it's because it's part of the beetle family. And so that hard casing, the spots on either side are actually very symmetrical. Actually, they're exactly symmetrical to each other. And so the hard shell actually is covering the wings inside. But I think first we're going to look at, so first we're going to try to zoom in to see if we can see the wings under the shell. So as we move and zoom in, your screen may look a little funny, but once we get to where we're going to stay, it should even out. And so we're zooming into basically the crack between the two parts of the ladybug's shell.
Right, so here's its shell, and then here underneath we're starting to see the wing right here.
And so, yeah, this, like Nicole said, this is the wing here. And actually, people study bugs' wings to figure out how old they are based off the amount of damage there is to the wings. And what you'll notice is that that wing had many fine features on it. So like little spikes and little hairs as well. And so those are things that we can't see with the human eye. They're only visible when we use these kinds of microscopes.
Right. And you'll notice a lot of this, there's just debris on the ladybug too. Dirt and things from the outdoors.
Right, yeah. I think so. And so now we're moving towards kind of the back end of the ladybug where we think we see one of its other wings sticking out as well. But it's a little too hard for us to see. So
this is the wing. Here we go.
Oh, okay. Okay.
Yeah.
So So also next to the ladybug, we put we actually took off one of her wings and put it down next to the ladybug, so we can give you guys an idea, a better view of the wings. And so sometimes we'll do this auto contrast brightness adjustment, and so that's why the screen looked really strange there.
Yeah.
Okay, so now you're seeing a really nice image of this wing. And you can really see kind of the fine surface features as well. And so you'll notice that there are some veins going through these wings. And scientists will actually use like how the veins look to trace back the ancestry of different bugs to kind of find out where they came from. And so we're going to take some pictures, too, if you want to come back in, for you guys as we go. And so you'll see the screen kind of do a slow, very slow movement across. And we will send you guys these pictures, too, so you can have them.
Okay. Are those hairs? It's hard to tell from here.
Yes. Oh, it is hard to tell? Yeah, those
Well,
are.
I see little white dashes, but I can't tell if they're raised up or
Yeah, those
toward.
are little tiny hairs on the wing. I know for other bugs that sometimes little tiny hairs are used as sensors, but since these are on the wing, I'm not quite sure what their function would be exactly.
Yeah, there we go.
So can you see those a little bit better now?
Yeah, I see those now.
Okay.
Good. And again, look down here at the scale bar. This is 50 microns, so you know the size of your hair.
Yeah, we're about looking at the size, the width of your hair right now in terms of length scale. So we'll take pictures as we go along for you guys, and I think next we're going to go to the eye. I think there are some interesting things to see there, and kind of as we look through all of these bugs, you're going to notice a lot of similarities. Same features, especially within the eye too. Okay. So bear with us for a second as we find the features.
It's fine.
I think actually, I put another ladybug next to this one that's upside down.
Oh, okay.
So I'm hoping maybe we can see the eye better.
So the first ladybug you looked at, like Nicole said, was right side up, and now we're going to go look at another one that we had actually. We look at ladybug samples a lot. So we've put one in there that's upside down as well.
Right, so right now you can tell it's blurry, so now I have to refocus, just like you would in a light microscope. And now we can start to see the hexagonal features of its eye.
And so you can't see these hexagonal lenses as you look at this bug, but these are pretty much common in many different bug species. And so these lenses actually allow the bug to see in multiple directions at the same time. And so as we look at other samples, you'll see these features too. So we're taking an image here. So you'll start to see the image come in a little bit better resolved as we do this slower scan.
Okay.
Do y'all have any questions, or do you want to look at another sample? Kind of, you guys call the shots.
Yeah, this is all you guys.
Questions about the ladybug? They don't have antenna, do they?
I don't believe so. If not from what I remember reading about them, I don't believe they do. But we can look at some other samples of you guys that you guys sent if you'd like.
Yeah.
Fly,
Sure.
bumblebee,
or the orange bug.
I think
All
the
right.
fly is a really good one. Need a recommendation.
One is
The fly.
the fly. Okay.
Yeah.
Okay.
Okay.
So we will go over to the fly. Mm-hmm. Oh, so Carrie, our fact checker in the back, just looked it up, and ladybugs do have antennae. So my guess is that they fell off on these samples.
Okay.
Okay, so right now you're kind of seeing a really creepy looking image of a fly.
Mm-hmm. So up here we have its head, right, and we have its eyes, one, and then another one right here, and then one of its arms coming out, and then this is its body. And so these are common house flies that you probably have had in your house.
Yeah.
All the time. And what you'll notice is they're going to be very, they have a lot of hair on them.
Mm-hmm. So where do you guys want to zoom in on? What looks interesting?
Its eye, its leg, its...
Somebody said the arm.
The arm? All right. We'll go there. And so what you see, it's got a lot of hair.
Yeah.
And actually, these hairs serve as taste buds. So whenever you've had a fly kind of like come chill out on your food, he walks around for a while. And that's because he's trying to get a taste of the meal that he could have through these little feelers on its leg.
Mm-hmm.
And there's a lot of them.
Yeah. And there's different types of hairs too. There's ones that are like this, and you have these ones which, when I zoom in, they have this microstructure to them, which I think looks sort of like a churro.
Yeah. So let's take a picture of this one, and then we're gonna zoom in on one of these churro-looking
Churro-like hairs. Yeah.
churro hairs.
Anybody want a churro later?
Alright, so we'll zoom in on one of those so you can see those features better. I'll focus.
Okay. So you should be able to see.
Can you all see that?
Oh yeah.
Yeah. Okay. So we got churro hairs here.
And then even beneath these larger hairs, you can see all these much smaller ones as well.
Yeah, the finer hair along the body.
Hair, yeah. So I'll zip back out.
Are those? Okay.
What was that?
Were those teeny little hairs at the bottom?
Yeah.
The
Yeah.
little white.
Yeah.
The little white tiny hairs.
Let me zoom back in. These down here.
They just the whole fly is covered in these, so these probably serve more as like a protective coating of the fly's skin. But the churro-like hairs are the sensors, the ones that can actually sense the taste of the food that they walk along.
Yeah.
Okay.
There seems to be some.
Oh yeah.
Okay.
Um,
All right. So that's the arm.
so where do y'all want to look at next? We got the eye. We can look along the body.
Any preferences?
What do you say?
So it's more like that head part.
Yeah. The head part? The hair is like around his neck.
Okay. Yeah, we can look at that.
It looks like he has a mane of hair in his neck.
Yeah.
Yeah, so this is a nice picture here because you can see the lenses on the eye once we adjust here, and you can see kind of the mane it's got going on back here.
Mm-hmm.
And it's a little dark just because our imaging is limited a little bit, but we're going to try to fix that. There we go.
Yeah. How does that look? You guys see that?
Yeah, that's good.
Okay. So just like with any kind of digital image, we can adjust the contrast and brightness of our samples as well. And so that's what we did in order for y'all to see that. And so we're going to...
At the very top.
Sorry?
That's where it looks like at the very top, it looks like an antenna fell off.
Oh, yeah. So, yeah, we saw a lot of places on this fly's head, yeah, where you're pointing out as well, where there were antennae, but they have probably fallen off.
Mm-hmm.
But the flies kind of have multiple antennae, and we're going to see if we can... Oh, so actually, right in the center here,
Mm-hmm.
if you see those three little bumps, can y'all see those?
Yes.
2, 3.
So those are what's called the simple eyes, or the ocelli, of the fly. And so not only do the flies have compound eyes with the hexagonal lenses, but they have these simple eyes that kind of serve more as like a compass, so they know which way is up. And so how these eyes do that is they go, they're sensitive to light. And so have you guys kind of noticed when flies are in your house, they tend to stay by the windows or by where there's light? That's like they're trying to figure out which way is up through those through the light that they can sense with these simple eyes here. So we're going to zoom in and get a couple images of this for you guys. And so the structure of these smaller eyes is very different from the compound eye. We don't have that hexagonal shape. And that kind of illustrates that they serve a different purpose too. And we got some churros going on in the back too.
Yeah.
Yeah.
And I think we'll take a quick look at their main eyes, just so you could see how they compare and are similar to the ladybug eye.
Yeah, so again, you're seeing tons and tons of these hexagonal lenses, and flies actually have 3,000 to 6,000 of these per eye, so there's a lot. At one point, you can kind of see some little hairs sticking out between the eyes. Typically, flies that are alive have all of these, so there's a hair kind of in every corner, but it's lost many of them. So.
Well, I think I found this fly. Nobody brought in a fly, did they? This fly was in my garage. It might have been there for who knows.
Okay.
And it was caught in like an old cobweb, so it could have been there for months or.
Oh. So we were wondering because we saw a lot of like cobwebby material all over him when we were doing the analysis, and we were wondering what kind of sadness.
And that orange bug.
Yeah.
Yeah, the orange bug was from the same area. As
okay,
well.
Yeah. That makes more sense now. And so flies actually live for about 28 days, unfortunately. Their lifespan is really short. But again, people can kind of, based off the amount of damage to the body, kind of gauge how old flies are. I'm not actually sure, since I'm not a fly expert, I can tell you how old this bug was before he died. But that's how people will age bugs.
Yeah. Okay.
So I think for this sample, unless there's anything else you guys want to look at along the fly's body, we can look a little more around here, or we can go look at the bumblebee or the orange bug.
Before we go on, were the wings gone on this?
We do have a wing on the side that we can look at.
We do have a wing that is sitting on the side to this, if y'all want to look at that.
Yeah, briefly, I guess.
Okay.
Nobody else seemed.
Okay.
Okay, so we're going to find it real quick for you guys. Okay, so it's on your left. We're coming into it here.
I'm trying to back up a little bit so you can. Right, so here's the bottom where it would be attached to.
That's a little better.
No.
We're just trying to make sure the image is the best it can be.
See it. Okay, that's better. So can you see this?
Yeah, I see it.
Okay. Right. So
the wing is.
it would be attached to the body, and then up here is the full wing. So we'll zoom in on this so you can see what the structure looks like.
So wings for bugs are mostly made out of chitin, and And so chitin, is that what makes up human hair? I believe humans also have body parts that are made up of chitin, but Carrie's going to fact check me here.
Is that fingernail?
Yeah, I think the fingernail, and I thought hair, but I'm not sure. So, yeah, I think it is fingernail, though. And so, again, you guys also, do you see all the little white dashes or hairs?
Yes.
Yeah, so it's pretty cool to see how similar, even though, you know, a ladybug compared to a fly, they are different kind of bugs, but they have very similar surface structure. So these little hairs here. And this long white thing in the wing is actually a piece of that cobweb. So
Yeah.
So we'll take a picture of that.
I don't know if a spider ate this at all.
Well, they use the sucky.
Okay,
Could
so
you tell if this?
Sorry?
Could you tell if this fly had got? Could you tell if this fly had ever met the spider?
had ever
Meaning, if a spider had found this fly, wouldn't it be more destroyed than it is? And that it was eating.
Yeah. I don't think it would be this intact if it had an encounter with a spider. Because I
think the spider.
Sorry.
I was just telling Matthew that I think the spider web is probably empty at the time.
Oh yeah. Yeah, he wasn't that unlucky. So now, is this the bumblebee?
No, this is the fly.
Oh, okay.
Yeah.
So now we can keep looking at the fly, or we also have the bumblebee or the orange bug.
We'll do
Orange.
whichever one's closer. I want to see both of them.
Okay.
Well, we'll look at both.
Okay, we'll look at both of them. So which one are we?
This is bumblebee.
Okay, so we're going to go to the bumblebee first just because it's situated the closest. And we'll adjust this image as well so you guys can see it a little better. Okay.
So what you're looking at now is we're looking at its face head on.
And so we have an eye up there at the top, and then its other eye is at the bottom.
Oh.
Yeah. And so you can see kind of all of the, yeah, there's an eye down there, and you can see all the cobweb too. Did you find this one in a cobweb?
I didn't get the bumblebee. You brought in the bumblebee. So where'd you find the bumblebee? On the ground? Outside? It was found outside on the ground.
Oh,
Okay.
Okay. Yeah, I'm not sure what this material is covering it, but so right from this image you can see both of its eyes, but then it also has kind of the center eye again as well. So again, these center eyes again are the ocelli, and they'll detect the changes in light intensity. So fun fact about bumblebees, they can't see the same colors that humans do. And so humans can see, you know, blue, orange, yellow, green, red. Bumblebees can see those colors, but they can't see red, but they can see in UV light. So UV light is the kind of light, the harmful rays from the sun. But UV light, there's also, on flowers, they'll have different patterns on them that can be visualized with UV light. And that's how bumblebees kind of detect and visualize what kind of flowers that they're interested in going to. And so, what is this?
His eye.
Oh, okay. The ocelli?
The ocelli. Yeah.
Okay, so this is one of his ocelli eyes here, surrounded by all of these hairs. And so why do you guys think that a bumblebee would be very, very hairy Like, what purpose does that serve?
Sydney, what do you think?
To carry pollen from flowers.
Yeah.
To carry pollen from flowers.
Yeah, that's exactly right. And so we know that bumblebees, their main kind of job in life, as well as, you know, making sure their species survives, is to carry pollen around to pollinate different flowers. And so bumblebees, they actually have these things called pollen baskets, which I never knew about until we started looking at these. But these pollen baskets can hold about 1 million pollen particles. So that's a lot. And so I'm not sure if we'll be able to see the pollen basket. They do fall off pretty easily, but we'll try to look at it when we look at the legs. And so this is just kind of a view of all of its hair. So you noticed it was cross-linked and kind of intertwined, and that really allows it to hold on to as much pollen as it can.
Look like the hair is branched off, too? Like one hair would branch.
Oh, yeah.
Yeah, it did look like that.
Yeah, we can try to zoom in on another area to see if that's consistent throughout.
Yeah.
And so this is kind of right by where one of its antennae is. And so, yeah, you do see kind of that branching structure of the hair, which is pretty cool.
I've never seen that before. See these branches here?
So we're just grabbing an image of that for you guys. Then on the bumblebee, we can also look some more at its head. We can look at an antenna. We also have one of the bumblebee wings next to it as well. Or we can go over to the orange bug. Your class ends at 11:05, is that correct?
11:10.
11:10. Okay. So yeah, we've got some time to look at anything else on this or the orange bug.
People are whispering the stinger. Would the stinger possibly be visible?
So interestingly, for bumblebees, the stinger actually comes off when they sting, for most of them. And that's how the bumblebee dies. So we didn't see a stinger on this one, and so I'm guessing that it stung something, and then when it pulls away after it stung someone, some of its insides come out with the stinger, and that's ultimately what causes the bumblebee to die. And among bumblebees, some will have much smooth stingers, and so some bumblebees can actually sting something and then get its stinger back out and stay alive. But other bumblebees have stingers that are barbed, and it's much easier for them to get caught up in the skin and then lose that stinger and eventually die. So, based off the fact we can't see a stinger, I'm guessing it lost it stinging something.
Yeah.
Yeah. Not really. Well, so bumblebees also, they do have some features around their mouth, which we're looking at on the right here. So a fly has kind of that plunger mouth, and bumblebees also have like a kind of plunger piece as well. It might have fallen off. We're going to see if we can find it.
Yeah.
You think we see it?
I know. There's a lot going on in this mouth area.
Actually, oh, this is its mouth.
Yeah.
Yeah, so we can definitely see one of its legs at the top there. And a wing as well. But the mouthpiece might have broken off, is what we think.
Yeah.
So, what do you guys want to look some more around the bumblebee, or do you want to go look at the orange bug?
I want to see the orange bug.
Okay.
So do they.
we'll go to the orange bug. So when we first saw this, we screamed.
Yeah.
Because it looked...
Not what was in the bag. Did you?
Where did you find this? In your garage?
Yeah.
Yeah? Okay.
Yeah.
So I did
It
a
looked
little...
really.
It what?
I said I thought it looked very cool, so I had to get it.
Yeah. Yeah, no, we got over our initial fear. So
You
it...
didn't scream when it was still in the bag, did you?
It what?
You didn't scream when it was still in the plastic bag, did you?
Oh no, we did.
We did.
Yeah.
Nicole showed it to me, and I was like, okay, I guess we got to look at it because it looks so strange and scary.
Yeah. So what you're looking at right here is its head.
Yeah. So.
And then back here is its body, which we're only seeing half of its body now. The rest of it is over here, but.
Is this on its side right now?
It is on its side. Yeah.
So he has a really small head, and I kind of Googled around to figure out if I could determine what kind of bug this was, and I think it's something called an assassin bug. So they can come in all kinds of colors and like different features, so it's really hard to say for sure. But these assassin bugs prey on other bugs. And so since, a garage is like a good place to find many different kinds of bugs, I'm thinking, based off where you found it, this could be an assassin bug. And so I believe there's this little, is that the eye there?
Yeah.
So if you see the little round thing on its head there, that's its eye. And so one thing that's different from the other bugs we looked at is it's not actually fully encapsulated by the body, but it sticks out. So that's something interesting. And again, all of these stringy things is probably the cobweb material. So we're going to focus on that. And so you don't see, you can kind of see maybe hints of the hexagonal lenses on this eye, but they're much harder to get in focus. And so they're not as prominent, I guess, in the other bugs that we've looked at. And then it's got these little, like, feeler things.
Yeah.
This is really creeping me out. It's got these, like, feelers, to kind of, I guess, sense what's surrounding it at the front here. So you can kind of see two, like, column features to it. Yeah. So there's two features.
And those went straight out from its head, like away from its body?
Yeah.
Yeah, so, they're extending out.
And they're very long.
They're about maybe 1 millimeter long.
Yeah, millimeter, 2 millimeter.
Yeah. So they're pretty long for a bug. So we're going to move over to the rest of its body. And it does have wings. So that's one of the kind of characteristics I looked for as I tried to identify it. And so you can see really prominent veins in the wings here. And then let's zoom in and see if we can see a similar microstructure or not. And so the wing actually looks a little more damaged. Like if you can see kind of it kind of looks like mountain ranges along it, if you see that. And so I'm guessing this bug may have been dead there for a while in your garage.
Could have been.
But we do see those finer hairs if we zoom in quite a bit. And so our scale bar here is about 100 microns. and these hairs are probably about maybe 1 micron. So that's 1/100th of the width of a human hair. So this is really, really, really small. So we're going to take a picture of that. Do we have any questions from you guys?
No? No.
Okay. So we're going to look a little more around this bug's body. So as we zoom in and out, our contrast and brightness changes, and so that's why we have to keep adjusting like we do.
Right.
So again, its head and its main body was over here, and here's its back.
I should have taken a picture of this bug because the students didn't even get this before we sent it off.
Oh.
Oh, they didn't?
You didn't take a picture of it, did you?
No, I didn't, but I can send you, I found a picture online that looks exactly like him. Or we can take a picture, well, yeah, we can take a picture when we take him out and send it to you.
Okay. Now when you prepare them, aren't they like coated in a very fine layer of some metal?
Yeah, so to make them not charge, basically, so we're shooting electrons at them, and we don't want them to just hold on to those electrons and charge up, because otherwise it looks like the image gets brighter. We coat them in a very thin layer of metal that basically helps make it look, helps it not build up that charge. So he may look a little bit gold to you when we send a picture, but you should still be able to see the orange and stuff like that. And so what we're looking at now is kind of zoomed in on his body. And so there's a lot of things going on here. We have like some round debris and again all the cobwebs too. And so bugs just pick up debris kind of either as they are dead or as they're alive as well.
Yeah.
So we're getting a picture of that. And then again, these tall, spiky features are hairs or feelers along the bug as well. And so I think we're just almost about out of time. So we've really enjoyed looking at y'all's samples, even though they did scare us initially a little bit.
Sorry.
No, it's okay. And yeah, we're really thankful that y'all were able to do this with us. So are there any last questions?
Any other questions? Have a seat, please. I'm dismissing you.
Oh, yeah.
Sydney has a question.
Yeah.
The copper tape that's on the stage, do you like reuse that when you put it, like, can you reuse the pages like put in there, or if you can reuse, but do you take the tape off and put new tape on, or how do you work that?
Okay.
Did you hear that?
Yeah. So basically, can we reuse that copper tape, or do we put on a new piece every time?
Yes.
So personally, it's kind of up to the person running their samples, but it kind of will lose its stickiness over time. And so when I look at samples, I do put a new piece on every time I look at one. It also like eliminates any chance for contamination from sample to sample too. So yeah, that's a good question though.
Any other questions?
One more?
Well, thank you very much.
Oh, thank you so
Yeah,
much.
thank you. It was a lot of fun.
Well, we appreciate it. We'll give
Yeah.
them a hand.
Thank you. All right.
I'm going to let them. You can go ahead.
All right, bye.
All right. Bye.